Hello, everyone, and welcome to today's webinar titled Advanced Solutions for mRNA Vaccine Development. This webinar is a part of the 6th Annual Bioprocessing Virtual Event Series 2024. I am Kaylee Bach, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. And with that, let's get started. I'd like to now present today's speaker, Feng-Mei P., Head of Product Development and Manufacturing at GenScript Innovation Center. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Feng-Mei, you may now begin your presentation.
Thank you for your introduction. I'm going to talk about vaccine development solutions at GenScript. Vaccine plays a very important role in our daily life and health. There are 3 generations of vaccine have been developed in the human's history. The first generation is a virus-based vaccine that includes both inactivated virus or live attenuated virus vaccine. Later, as a second generation, those are many called subunit vaccine, where we have extracted the antigen by the peptide or protein or format, and also there's a virus-like particles vaccine. And lately, the third generation of vaccine have been emerged, led by the COVID-19 vaccine. Those are nucleic acid-based vaccine, include the DNA or mRNA vaccine. They can be delivered in either LNP format for mRNA vaccine or virus vectors for the DNA vaccine. By looking at the global infectious vaccine research and development, the recombinant protein vaccine ranked first, but nucleic acid vaccine come close to the second. Here is a landscape of the infectious disease vaccine published in Nature from last year. We want to take a moment to look at the mechanism of how vaccine works in life. So first, the vaccine is got to be introduced into cells. The vaccine in either the DNA, mRNA, or protein format are get transfected into cells using their delivery vehicles. And for nucleic acid-based vaccine, they get translated into the protein format and then presented into the T cells for its expression and stimulating the immune reactions. And for virus vector or the peptide protein-based antigen, they will be presented by MHC molecules on the dendritic cells, then activates T cells. Then the CD4 T cells drive B cells development, results in maturation of antibody response, antibody-mediated neutralization for immune protection effect, while the CD8 positive effector T cells are important for the elimination of infected cells. Next, I want to introduce about the vaccine development solutions we could offer from GenScript. At GenScript's life science group, we have the following services from gene synthesis, mRNA and RNA-based oligo synthesis, and peptide synthesis service, protein production, antibody production, and also research reagents business units that provide solutions for your vaccine development projects. That covers from the basic research, vaccine development, diagnosis, and other gene cell therapy applications solutions. The vaccine development solution from end to end, we covered from mainly the early phase. At the very early stage, antigen discovery. So we will provide solution for antigen identification and selection that include the pathogen study using genome sequencing method by qPCR probes. We have provide this primer synthesis service, and we have the oligo pool synthesis service using Gene Titan silicon-based chip oligo synthesis technology, and also NGS targeted enrichment solutions, and other PCR reagents. We also have solutions about protein expression service, protein purification service, protein electroporation, and Western blot studies support, and antibody development and labeling service. We also have the peptides library services in our peptide department. That also provide a tool for your neoantigen peptide tumor study by neoantigen peptide synthesis. Moving on, in your early phase of the vaccine development, you can develop either like DNA for either DNA-based vaccine, mRNA-based vaccine, or peptide vaccine. We all offer those specific synthesis service. For DNA, we have gene synthesis service, plasmid preparation, AAV vector one-stop solution, and an antiviral vector one-stop solutions. For mRNA vaccine, we offer IVT mRNA synthesis and the novel type of circular RNA synthesis and self-amplifying mRNA synthesis service. We also have the LNP formulation for mRNA that can directly formulate the mRNA into ready-to-use format for your research project. The recombinant protein vaccine, we have insect expression, mammalian CHO expression services. And we also have peptide synthesis service, peptide vaccine solution by neoantigen peptide synthesis, and regular peptide synthesis service. Moving on to preclinical studies, in the antigen expression experiment, our product development has solutions of precast gels, the eBlot protein transfer system, which is a very handy electroporation system that make it more efficient comparing to traditional wet blotting system, and easy west blotting devices. Humoral immunity evaluation service, we offer the ELISA kits generation service, the SARS-CoV-2 based pseudovirus neutralization assay, and the CPAS SARS-CoV-2 neutralization antibody detection kit. For cell immunity evaluation, we offer custom peptide pool synthesis, antibody development, and labeling research service. At the later stage, our sister company, ProBio, provides more GMP-based plasmid DNA synthesis and GMP scale peptide synthesis and GMP biologics. We also offer service for customized ELISA kits development, endotoxin detection kits, and lentivirus P24 ELISA kits, AAV2 ELISA kits, BSA ELISA kits, and other nuclease and ELISA kit development service for your research project. So autogene covamin is an investigational mRNA vaccine co-developed by BioNTech and Genentech as an adjuvant therapy in cancer treatment to prevent cancer recurrence following surgery. Autogene Cuvermin is a type of individualized antigen-specific immunotherapy containing multiple different mRNA species that together encode up to 20 new antigens have been identified in the resected tumor of a patient. The aim of the vaccine was to stimulate the immune system to mount adaptive response to any remaining cancer cells with these mutations. So this publication mentioned that using our company's service of neopeptide synthesis, they got very good progress and now it's in clinical trial phase. Next, let's move on to take a look about vaccine development solutions in more detail. Our gene synthesis service has about 20 years experience, and we committed to quality and dedicated to innovation. We are imaged with the trust and the love from our customers, and we have top-notch quality assurance, and with a lot of experience of delivering more than 3 million genes in the past years with more than 99.9% success rate. We also offer the GenSmart codon optimization service, which have a lot of citations showing that outperform the other algorithms. And we offer the GenBreak gene synthesis up to 200 kb DNA synthesis service. We also have the VectorArk and the CloneArk service that for you to storage your plasmid and constructs with us for easier material and data management. They're in Doctor newspaper about the published in Nature Biotechnology in 2018 about prime editing. They have found that GenScript's codon optimization alone outperformed others by inducing 1.8 higher fold editing efficiency. So this GenSmart algorithm is free online, and you can use when during your experimental design phase. We also got feedback from another customer, Novavax, who is a leader in the SARS-CoV-2 vaccine development, and they have mentioned that GenScript synthesized the SARS-CoV-2 spike protein that in 3 days that was very remarkable and speed up their research projects. So our plasmid preparation at GenScript include different grades. For basic research, you could choose our research grades, plasmid DNA, and the early phase drug discovery phase. Industrial grade might be a good solution, and the preclinical studies, the preclinical grade is also available. So that include our default QC and add-on QCs. We guarantee highly about the low endotoxin. We keep all the endotoxin level at less than 10 EU per mg, and we also make sure there is no residual RNA products, and residual host protein detection. The add-on QC will include advanced endotoxin removal and mycoplasma contamination test. Our preclinical grade plasmid preparation are ideal for preclinical in vivo animal studies from mice to non-human primates that include a very comprehensive and stringent QC and tailored for preclinical studies, and they are available in the quantities of 10 milligram or more. Moving on at the GMP stage, we also offer GMP-grade plasmid synthesis at GenScript ProBio, where the ProBio has a lot of experience to service the pharmaceutical companies developing their vaccine products. Next, mRNA used our service for the COVID-19 mRNA vaccine development. Abogene is also have a long history with us using the first round COVID-19 project setup. Next, moving on, we also have the mRNA vaccine service that include from gene to mRNA and LNP one-stop solution, where you send us your ORF sequences. We could help you to do codon optimization, and then start there. We do gene synthesis, clone it into our pre-validated mRNA vaccine vector, and then we do IVT to prepare mRNA or circular RNA or self-amplifying RNA. At the end, we load it into the LNP format for your studies in vitro or in vivo. By the way, we also provide solution of the enzyme production, which are used in the IVT process and the RNA production process, like T7 polymerase, RNase inhibitor, DNase 1, protease K, and other enzymes like RNase are used in circular RNA production. So our customized mRNA service workflow is like this. You could send us your ORF sequences, and we will help to design that with the codon optimization and clone it into our mRNA expression vector. And moving on, we'll start with gene synthesis work and the plasmid preparation. And then the plasmid will be sent to our mRNA production center, which is the innovation center in Seattle. We will do the linearization and IVT to prepare your mRNA. And the mRNA can be finished with standard QC or advanced QC at your choice. So for mRNA, there are two critical attributes that would impact the mRNA's quality. One is the integrity, and the other is the poly-A tail integrity. So for the improving the mRNA integrity, we have optimized our IVT system to generate the mRNA with the highest integrity and lowest degradants or double-strand RNA byproducts. You can see by comparing to competitors, we have slightly better integrity by bioanalyzer analysis, which is showing on the top left. But this will translate to very importantly about the expression intensity. We can see our mRNA has much higher expression in cells. And a second factor about the mRNA quality is the poly-A tail. Poly-A impacts about the mRNA stability. We recommend to build the poly-A tail into the plasmid during the plasmid design and preparation stage. By then, we can get a good mRNA product with very uniform poly-A tail. So this top right corner is a poly-A tail assay comparing different vendors' poly-A tail quality. So vendor A's trace is a typical character of poly-A added by poly-A polymerase, which has a flat distribution, and that translates to lower expression efficiency. Poly-A tail solutions. Here we want to share a little bit more about the details. We studied how the length of the poly-A tail impacts mRNA expression. In this experiment, we added the poly-A tail into the templates by PCR using gene fragments product as template, and then did IVT to prepare the mRNA, and then transfected this mRNA into A549 cells using lipofectomy, and then look at its expression after 24 hours. So the poly-A length did play an important role. The longer poly-A length, it get better expression. Overall, we would suggest more than 80A is better. But 100A is our standard format in our vectors. Regarding to poly A, we also offer the poly A, the oligo DT purification, which is an affinity purification that help you to get a further higher purity of RNA. This is a solution I would recommend for you when you have mRNA that is longer than 4 or 5 kb. Your RNA by performing this purification could greatly improve your RNA purity. And regarding QC, we also offer the poly A tail length assay using an enzymatical cut followed by LCMS. With that, we will give you the exact distribution of the poly A tail. So usually we see a distribution for our 108 service. We get from 108 to 111 A's in the final product with even distribution. More QC, we have developed a very comprehensive QC spectrum for the mRNA. Some useful or add-ons that you could consider for your RNA vaccine development. Those include the capping efficiency assay and double-strand RNA residue assay and the protein residue assay, since those could impact a lot about the RNA's immunotoxicity and efficacy. So capping efficiency assay, we use RNase degradation followed with LCMS. We have confirmed our capping efficiency is more than 97%, but we could provide this service for you for your mRNA or mRNA synthesis with us. Double-strand RNA residue assay, we use a immunoblot-based method where we have optimized and could provide a quantitative number for you at the end, and the double-strand RNA residue numbers will be usually less than 1%. Definitely less than 1%. We usually control it's more around 0.2%. So Cap2 mRNA is a novel type of RNA that only provided at GenScript. So Cap2 RNA actually is a natural occurring RNA type that's has a reason for it to be exist in the human cells, should play an important role. And there is research showing that Cap2 RNA has a higher expression at immune compromised cells conditions. So here we also observed that when we challenge the cells with interferon alpha, we observed the Cap2 RNA outperformed the Cap1 in its expression efficiency. We are the only provider that could do in vitro synthesis of Cap2 mRNA using co-transcriptional capping technology, which is our patent pending technology. And the scale we could offer is 0.1 to 5 mg, and we use the ACA-Cap2 cap analog for your synthesis, and you could choose the N1-methyl pseudo-U or 5-methoxy-U pseudouridine or 5-methyl pseudo-U modifications. And we offer the similar QC assays for the Cap2 mRNA based on your research needs. Moving on, the circular RNA is a more novel type of RNA type that is considered as the next generation of mRNA, which instead of 5' end and 3' end of linear mRNA, this is a covalent link and form a circle, which can resist to exonuclease degradation and provide better in cells stability. So our service include a scale of synthesis from 0.2 to 10 mg of circular RNA synthesis, and the length limits so far is less than 5 kb of the whole circular RNA, and we provide RES options. Currently, CVB3 is a default, and we have more RES to offer and the data to share with you later. The purification, we would recommend using our optimized proprietary HPLC purification, which could get a high purity product, which correlates to its expression efficiency importantly. And The QC assay for the circular RNA include identification test. Usually we run agarose gel, and there's add-on RNase R degradation assay, and we further can offer the add-on purity assay by reverse phase HPLC, and we also offer the QC for the impurity test for plasmid DNA residue, protein residue, and endotoxin level assay. So circular RNA by structure-wise, since it doesn't have a cap, it need to use the for coding RNAs. We included an IRES section, which help to recruit the ribosome and help with the translation of IRES-dependent ribosome recruiting. So the final product will include RES, your ORF sequence, and there is a scar from the permuted intron exon reaction will have some exons and adapter sequence in your final circular RNA product. As GenScript, we have so circular RNA production, one challenge is how to make a good purity and the scale. So we have our IP pending technology where have our special design that help to improve the circularization efficiency. In return, it will translate to higher purity after proprietary HPLC purification. We did more study to look at how the expression of circular RNA comparing to linear mRNA. So at both protein or RNA level, during 3 days time course, we observed about threefold higher expression of circular RNA compared to linear mRNA. And we also look at the half-life of the RNA copy numbers by qPCR. You could see on the right graph, after transfecting equal molar amount of RNA into cells, the circular RNA copy numbers was about 2 to 3-fold higher compared to linear RNA after 24 hours. And this trend continues, and you can observe about generally 1 to 2-fold higher of RNA copy numbers in circular RNA format versus linear RNA format. Moving on, RES is one important component in circular RNA design. So the CVB3 is a widely validated RES that could used in your circular RNA vaccine development, but different cell types might have different preference of the RES. So here we did a screening, and these 4 are the winners that have higher circular RNA expression in different cell types. That could be a good reference for your circular RNA product development. Next, self-amplifying mRNA is also a good solution for vaccine development since it can have the replicants in the RNA design, which can greatly reduce the dosage requirement and extend its half-life in the cells. So we have an optimized self-amplifying mRNA vector for your self-amplifying RNA design. You could send us your gene of interest and we could put it into our vector, or we could do it with your design. The scale we can offer is anything from 0.1 to 100 milligram, and the circular RNA usually has a cap1 structure. We could do it through co-transcriptional capping method, and in this vector we have 60A by default, but it can be customized as well. Available modifications by default is unmodified. We have also found that 5-methylcytosine modification is compatible with self-amplifying mRNA for your study. And the purification method, by default, we use lithium chloride precipitation, and there is upgrade using oligo-DT purification. So a quick summary about the difference between all the among the 3 formats of RNAs: linear, self-amplifying, of circular. So comparing about protein expression, uh, linear mRNA give you very strong strong protein expression, and which is also a very mature platform that's quite reliable. And the self-amplifying RNA gives strongest, and the circular RNA gives stronger protein expression. And the half-life wise, due to the self-amplification nature, so the self-amplifying RNA has the longest expression duration, followed by circular RNA and linear mRNA. But immunogenicity-wise, linear mRNA has the most optimized and lowest immunogenicity. You would expect some medium to high level of immunogenicity for the circular RNA and the self-amplifying mRNA. Um, in return, the dosage requirement for mRNA might be higher, and the requirement for for the circular RNA and self-amplifying mRNA are lower. So on the right corner, there's data we did in vitro look at the protein expression after transfecting 200 nanogram of EGFP RNA in linear format or circular RNA format or self-amplifying RNA format. We could see the expression difference of the three type of format. Self-amplifying RNA gives the strongest expression, followed by circular RNA and then linear RNA. Yeah, last and not least, we offer the LNP formulation service, which can be used to encapsulate your RNA vaccine into LNP formulations. Um, we use the generic ionizable lipid formulations, and we have four well-validated formulation. Those are MC3, SM102, ALC03, or LP01 based LNP formulations. We could load any scale from 1 to 30 meg of RNA, and the length can be anything from 100 nucleotide to 15 kb of RNA. We have validated this data, this platform by injecting the LNP into mice by intramuscular injection or IV injection, and they are also suitable for certain in vitro studies, especially for T cell transfection. The LNP will be finally delivered in a liquid format and will ship it on dry ice. And we suggest to limit the freeze-thaw cycles for the LNP formulation. Um, the standard QC for the LNP will include the encapsulation efficiency, where we guarantee more than 85%, and the DLS assay to measure its size distribution, polydispersity index, and zeta potential will ensure a very uniform LNP formulation with less than 0.2 PDI. And the pH will be around 7.54, and the endotoxin level is always less than 4 EU per ml. The final concentration is an optional choice that we can go up to 2 mg per ml of the LNP formulation. So here's some data that we want to share with you for MC3 LNP encapsulated with fluke mRNA, and we injected into mice by intramuscular at a dose about 5 microgram per mice. We observed a very strong expression of the protein after 48 hours, and it outperformed other two competitors' formulations, and it has good expression, mainly accumulated in liver as LNP. And we found it's quite safe during the short time course of the study. There's no toxicity was observed. So overall, for the mRNA vaccine development, you might consider about all these factors: plasmid DNA design, the mRNA integrity IVT conditions, and how the poly-A tail quality want to build, and the mRNA capping solution modifications, and the impurities, and also lipid LNP formulation. And so we will provide you a one-stop solution for the mRNA vaccine study, from gene synthesis, from the codon optimization to final LNP formulation, where we have our proprietary vector and proprietary circular RNA vector and IVT method for co-transcriptional capping of Cap1 mRNA, and also advanced purification solutions, including oligo-DT and double-strand RNA removal for the RNA product. Moving on, at GenScript, we also offer vaccine effect evaluation service for your preclinical and clinical studies. We have the ELISA kit generation service for humoral immunity evaluation. So serologically tested of IgG M antibodies and against the antigen. And we also tested the neutralizing antibodies against the virus in the CPAS product. Here's some feedback I want to share with you. That GenScript and Biocom joined forces to accelerate novel intranasal SARS-CoV-2 vaccine development in Japan. We also have feedback from customer who developed the circular RNA vaccine against the SARS-CoV-2, used our CPAS SARS-CoV-2 serological virus neutralization test kits and got very good results. We are very proud to provide all the solutions for your vaccine development projects. Please let us know if anything we could help with you. Thank you very much.
All right. Well, thank you, Fengmei, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located at the bottom of your screen. All right, so let's get started. Our first question here asks, how efficient is your plasmid linearization service, and what percentage of the plasmid is in linear format?
Thank you for the question. Yeah, so the plasmid linearization, usually we ensure it's more than 98%. It's quite very well validated service, because we know the quality of the linear plasmid is crucial for the RNA quality as well.
Great, thank you. All right, another question I have for you here. What is the most important parameter to optimize for mRNA stability?
Yeah, thank you for the question. That's a very good question. So there are several factors. Of course, the modification, usually with U modification will help to improve the stability. And secondly, what is the buffer used in the final storage? We have performed the stability study for longer mRNAs, I mean like longer than 5 kb. Usually preserving 1 millimolar sodium citrate will help to preserve the better stability. And also the sequence, the UTR sequence and the poly-A tail might also impact mRNA stability from the design.
Great, thank you. Now, is your timeline longer for producing circular mRNA compared to linear?
Yeah, thank you. So just in the part of the RNA production, the circular RNA timeline is a little longer. We need about 3 weeks, since it involves of a precursor circular RNA preparation and the circularization and then purification process than linear mRNA. But overall, the template for circular RNA usually easier to make, since it doesn't have a long poly-A tail. I would say from beginning to end, they have a similar timeline. We would look at around 6 to 7 weeks from gene synthesis to final RNA produced. Yeah, thank you.
Great, thank you. Well, it looks like we have time for one more question here, so we'll wrap up after this one. And this question asks, can you decorate lipid nanoparticles for stem cell targeted delivery?
Yeah, thank you for the question. Yes, for the LNP targeted delivery, we could decorate the LNP with antibodies, which is specific to stem cells, such as CD117 antibody. Yeah, it is doable. And we could provide that service. Thank you.
Great. Well, thank you again, Feng Mei, for your time today and your important research. Be sure to also check out GenScript's booth located in the exhibit hall for more great information. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 3rd, 2025. Labyrinths will alert you via email when it's available for replay, so we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.